Welcome to Catherine's tutorial. In this video, we learn about Messelson and Stahl's experiment. In our previous video, we were learning about Griffith experiment, transformation experiment, and followed by Avery, MacLeod and McCarthy experiment and Hershey and Chase experiment. All these experiments was done to prove what was the material carrying the information from the parent to the offspring, whether it is DNA or protein. But finally, they just come to a conclusion that DNA only carries the information. Okay, and here we are going to learn about how DNA replicates. There are three hypotheses have been proposed to explain how DNA replicates. We'll just see one by one. There are three hypotheses been proposed to explain how DNA replicates. The first one is known as conservative method. Conservative method of replication. So according to this method, the DNA, that is a double helical structure, when it replicates, according to conservative method, when DNA replicates and produces a copy, the parental strand has been kept as such. There will be no changes in the parental strand, but the new strand, that is a entirely new strand or a daughter strand is formed as a result of replication. This was conservative method. So this is a parental strand. This is the parental strand which was kept exactly as such, and this is a new strand has been formed. This is how the conservative method explains how DNA replicates. The second method is nothing but the semi-conservative method. What is semi-conservative method? This method was proposed by Watson and Crick. It was proposed by Watson and Crick in 1952. But when you take sign, the experimental evidences are needed to support any hypothesis. So there was an experiment conducted in the year 1958. 1958 by Messelson and Stahl to prove this semi-conservative method of replication. This is how the DNA gets replicated. So how the DNA gets replicated, we'll just see here. So this is the parental strand. According to them, during replication, one of the strand, that is one of the parental strand, acts as template, and the new strand has been formed, copying the nucleotides from the parental strand. So this is how the semi-conservative method explains. So this is the parental strand. So this is parent again. This is the new daughter strand. So according to them, when DNA gets replicated and they form the copies, one copy contains the parental strand, the other copy contains the daughter strand. The third method is known as dispersive method. Dispersive method. So what is this dispersive method? According to dispersive method, imagine it is a parental DNA strand. It gets into pieces, and each piece will produce a new strand, and these two new strands, they form a complete strand. They integrate and they form the new DNA. So this is how the DNA gets replicated. DNA gets replicated or incorporated with the new daughter strand and parental strand in case of dispersive method of replication. These are the three type of hypotheses have been proposed to explain how DNA replicates. But the most accepted method among all these things is nothing but the semi-conservative method, which was proposed by Watson and Crick around the year 1952. It was explained by Messelson and Stahl in the year 1958 by an experiment. Okay, we'll just see an experiment, how they have just proved the semi-conservative method is a correct method of replication. We'll just see how Messelson and Stahl done the experiment to explain the DNA replication process. So in science it is considered as most beautiful experiment. We'll just see about the most beautiful experiment in detail. So 1958, Messelson and Stahl. They have done the experiment to prove DNA is replicating by means of semi-conservative method. So what was the materials or what was the organism they have taken for the experiment is nothing but E. coli. And one more component they have taken, it is nothing but isotopes. These isotopes are not radioactive. So we have not taken any radioactive isotope. Instead we have taken the isotope which is N15 and the other one is N14. This is heavy nitrogen and this is light nitrogen. So based on the heaviness only, we have selected an isotope. Not we have selected, they have selected an isotope is N15 and N14 isotopes they have selected. Okay, so what they have done with these isotopes? So what they have done, they have taken a culture, two different cultures. So these two different cultures contain E. coli. And this E. coli was cultured with N15. This E. coli was cultured with N14. Okay. So when E. coli replicates, so the E. coli replicates in a laboratory condition within 20 minutes. They form a new daughter DNA strand. So what happens here? They need nitrogen as a source for their replication process. So they'll absorb nitrogen from the medium what we have provided. Here it is N15 and here it is N14. As we have learnt here, N15 is a heavy nitrogen and N14 is the light nitrogen. So they made a culture, they made a culture where the E. coli of one group will grow in N15 medium, the other group will grow in the N14 medium. So what they have done, they just made them to grow. Okay, this culture was happened around nearly for 14 generations. So they made this nitrogen to incorporate with the DNA of E. coli. So what may have happened? So this E. coli DNA may have incorporated this 14N into their DNA. So the DNA of E. coli contains N14 in their DNA and here the E. coli of this group contains N15 in their DNA strand. So it is incorporated, it has entered inside the DNA of E. coli, that is N15 has entered here and N14 has entered here. So what they have done like cesium chloride gradient centrifugation. Cesium chloride is a salt actually. This is salt which is around 17.1 grams in weight. The weight of cesium chloride and DNA is almost same. So it is the weight of DNA and this cesium chloride salt is almost same. So what they have done, they made a centrifugation with this E. coli and there is a separate centrifugation was done to this group of E. coli. So when they have done the centrifugation, they have collected this E. coli, they made an extract. This extract was taken for ultracentrifugation. So they have taken for ultracentrifugation process these components and these extracts were made to spin around nearly 140,000 times for 20 hours in ultracentrifugation process. So after ultracentrifugation, the materials inside the organism will settle according to their density. So what happened here, when they have taken the density from this particular group of E. coli alone, a band which is at the lower region. So it is heavy, so which settles at the bottom, which contains N15. So it tells you that N15 was incorporated inside this DNA of E. coli. And they made a centrifugation with a separate group of E. coli where you can able to find the bands of this particular group was found at the upper region. So this band was light because we have cultured this group of E. coli with a light nitrogen, this group of E. coli with heavy nitrogen. That is why this heavy nitrogen settles at the bottom and here the light nitrogen source that settles at the top of the centrifugation gradient. So this is heavy and this is light. Light contains N14 isotope and heavy contains N15 isotopes. Okay, 0. I'll write here. So these two are considered as generation 0 because there is no mixing of N14 with the N15 medium. But what happened in the generation 1? It is termed as generation 1. So what happened in generation 1? They made a group of E. coli which is under N15 medium. This is E. coli in N15 medium. This was provided with, this E. coli was cultured in N14 medium. It was cultured in N14 medium for 20 minutes. Extract, when they have taken for centrifugation with cesium chloride centrifugation or density based centrifugation process, they found the band which was in the intermediate. It is not light, it is not heavy, they have got intermediate band. This intermediate band is also known as hybrid. Okay, so this is considered as generation 1. The next generation was generation 2. What they have done in generation 2? They allow this group of E. coli. Okay, so imagine it is the first culture where we have got E. coli in N15 medium. These E. coli was transferred to N14 medium for 20 minutes. Again, this group of E. coli was taken, or when they kept again in N14 medium, it was around 40 minutes of time has been given for replication. So after 40 minutes, they took the extract. When they have taken for centrifugation, they found a band which was in the middle. One band was in the middle, the other band was in the light region. So this is light band and this is hybrid. Hybrid is also known as intermediate. Okay, what is this process explains you about how the semi-conservative method has happened. We have to compare with the DNA strand so that you can able to understand it better. We'll just compare this with DNA strand. In generation 0, okay, so what has happened? We have cultured E. coli in N15 medium and separated in N14 medium. We made them for centrifugation where we got heavy band for N15 group of E. coli and light band for this N14 group of E. coli. When you take this generation 1, it is the parental strand. It is the parent DNA. We made this parent DNA which contains N15 alone. We made them to culture with N14 isotopes. So what was the result we have got? One strand was the parent strand. It separates. The parent strand separates and unwinds and forms two separate strands, and the new strands are formed. These strands are known as daughter strands. So one parental strand which contains N15, the other strand is new strand, that is daughter strand, which contains N14. That is why we have got the band in intermediate length which has both N15 and N14 nitrogen, okay, which is not heavy, which is not light. It is a combination of both heavy and light in case of generation 1, okay. And when you compare generation 2, we made them to again to go for replication process of 40 minutes under again N14 medium. Imagine this strand again we have to take. This black color which is considered as parental strand which are divided again. This daughter strand also would have been uncoiled for replication process. So they uncoiled. So they separated to form the daughter bands. Okay. So what have happened here? This parental strand separates and they form a new band which will be with one is N15, this is N14, and they formed a new strand which is again N14 and N14. They form a new strand which is N15 and N14. Again the new strand was formed here which is containing N14 and N14. Okay. So as we all know, these light bands consist of N14 alone and these bands consist of a mixture of N15 and N14. That is why during generation 2, we have got one band in the hybrid, that is a combination of N15 and N14. The other one we have got in the upper region, which is nothing but N14 plus N14. Okay, so we have got the bands with the intermediate density. The other group of bands are remaining on the upper region, that shows the density is light. Okay, so based on the semi-conservative method, when we compare the generation with the DNA strand, we can able to come to a conclusion that when a parental DNA undergoes replication, one strand of the parent remains or acts as a template, the other strand will be a newly formed strand. That is not as a newly formed parent. One strand is a parental strand, the other strand is nothing but a newly formed daughter strand in case of semi-conservative method. By this way only the DNA gets replicated. But when we compare this generation 0, 1 and 2 with conservative method, what was the conservative method? The conservative method explains about the parental DNA remains as such when they form a new strand, and the entirely new strand or new daughter strand will be formed in the next generation. This was the conservative method. If conservative method was completely correct, one band should have been containing N15, the new band will be containing N14 medium, or if it is N14, we would have got the centrifugation result as one band will stay in the heavy region, the other band will stay in the light region. So we didn't get light intermediate band in case of conservative method. So this explains you that this method was wrong. If this method was correct, we would have got the bands in lighter region and also heavy region. But what we have got? We have got a bands in between, that is lighter region and hybrid band. So it tells you that this method was totally wrong. So this semi-conservative method was most accepted kind of method of replication in case of genetics or in case of DNA replication. By the next video, we'll learn about the next concept in genetics. Thank you.